Thank you for giving me the opportunity to speak and present Juventas. I will try to move through this quickly since I realize I am the barrier to lunch right now, so I'll get moving. Juventas is a gene therapy company that's building a non-viral platform to address regenerative medicine through advanced cardiovascular disease. We have licensed our technology from the Cleveland Clinic. We've moved it through phase 2 clinical studies in multiple indications and have demonstrated a strong safety signal with initial proof of concept, now focusing on peripheral artery disease, which I'll spend most of my time speaking about. The focus really of the company is to develop commercially viable therapies that activate or enhance endogenous stem cell-based repair to promote clinically meaningful outcomes. Essentially, we want to activate your own stem cells so that we don't need to take them out of you to redeliver them. It's a factor-based strategy to regenerative medicine. Our lead product is called stromal cell-derived factor 1, or SDF1. SDF1 and receptor CXCR4 have now been the subject of several thousand publications from several dozen labs and is understood to be the key axis for regulating stem cell-based repair of really any end organ system. We know through our research and that of others that SDF1 really works through multiple mechanisms. It was initially discovered as a homing factor through its ability to recruit endogenous stem cells to damaged tissue, both bone marrow-derived as well as tissue-specific. We know that SDF1 grows new blood vessels and that these blood vessels are sustained for long periods of time after the SDF1 signal is gone. We also know that the molecule has direct trophic effects and is able to prevent ongoing cell death. Together, what we've been able to see in multiple animal models and now clinical studies is that this helps prevent remodeling of scar and to help heal or preserve tissue after injury. So we've decided to exploit this system by introducing SDF1 to damaged tissue through the use of non-viral DNA. So this is gene therapy, but it's very different from the gene therapy that we've been typically hearing today. We are not looking for high transfection efficiencies and long-term expression. Rather, our goal is to get transient expression very local to the area of injury. Our plasmid has now been delivered to more than 200 patients. We've really seen no safety signals associated with either the plasmid or its delivery. Importantly, it's non-immunogenic, and this allows for repeat administration, a factor we thought was very important as we went after chronic progressive diseases, believing that a single administration of drug may not be sufficient and we need a modality would allow us to continue to reintroduce over time. I think equally importantly, when you think about the context of a commercially viable product, JVS100 is highly scalable and highly stable. We now have transferred this to 2 facilities with global manufacturing capabilities. It is generated through microbial cell banks, so no use of mammalian cells, and we have stability out to several years for the drug in a refrigerator setting. So that allows us to have nominal cost of goods and a easily distributable product from a logistics perspective. We have advanced JVS100 across multiple platforms. Really the take-home message from this slide is we do a really good job at helping heal tissue, heal muscle and skin. We know we grow blood vessels. We have clinical data in Ph.D., which I'll focus on more significantly since that's our most advanced program. We also have clinical proof of concept in heart failure and dermal scar. Just briefly, in heart failure, we ran a phase 2 clinical study in patients with advanced ischemic symptomatic heart failure. And what we were able to show in that study was through a pre-specified analysis that patients with high risk or advanced heart failure diagnosed as having very poor cardiac function had strong responses a year after delivery of JVS100 in really a number of different factors, including ejection fraction, size of the heart, and serum biomarkers. We have a phase 2B allowed by the FDA and received fast-track designation on that, and are looking to partner, raise more funds to move that forward at the right time. We also have some very interesting data in the area of cosmetic scar prevention or post-surgical healing. What we were able to show in this phase 1B study is that if you dermally apply JVS100 after surgery, in the case of this clinical study, a median sternotomy, you could accelerate the healing of that wound, you could reduce the total scar volume, and it had potential cosmetic benefits over time. Obviously, direction for this to go is in more cosmetic or plastic surgery indications, and we're having some discussions in that area as well. We have data also demonstrating preclinically in urinary continence and muscle regeneration that following injury we can preserve muscle, revascularize it, and improve contractility and function. But our main focus these days has been peripheral artery disease. Advanced Ph.D., also known as critical limb ischemia, is an area of great interest within the regenerative medicine field and a growing medical problem. There's an estimated 8 to 12 million patients in the United States with PAD. About 2 million of those progress to what's known as CLI, critical limb ischemia. The therapeutic options for these patients are very limited. For the most part, from a drug perspective, they're all preventative, helping or changing lifestyle, preventing the progression of disease. From a wound perspective, these patients tend to suffer with non-healing wounds, a lot of ischemic pain, and are at risk for amputation. A lot of this is standard wound care. The primary focus for these patients, our primary intervention is typically through device, whether that be stenting, angioplasty, or atherectomy. What we know in this population is that the goal is to replenish blood flow and help wounds heal. And what we know for JVS-100 through multiple studies is we do both of those very well. In a hind limb ischemia model of rabbits, what we're able to show is that when we inject JVS-100, after the injury, we significantly grow blood vessels. And we and others have shown the ability to heal dermal wounds, including those in diabetic models. And you can imagine a large portion of these patients happen to be diabetic. Based on our knowledge of the biology, we ran a phase 2A clinical study called STOP-CLI. Here we looked at 4 escalating doses of JVS-100 against placebo in patients that were classified as Rutherford 4, which has pain, or Rutherford 5, which has pain and non-healing wounds. In this study, all patients were poor revascularization candidates, meaning they'd exhausted the device interventions that were available. It was a small study primarily focused on safety and not powered for efficacy, but we did see several compelling trends. Specifically, a year after a single administration of drug, we saw trends in wound closure in our target therapeutic dose, and we also saw a reduction in pain, 2 key component clinical outcomes for these patients. That translated into improvements in Rutherford class and was supported by improved hemodynamics, or blood flow to the heel as measured by ABI. So phase 2A showed us that we had a safe therapy and one that had the potential impact of restoring blood flow and helping heal wounds. And those were key components to helping us structure the phase 2B study. Until recently, the CLI field is really focused on amputation-free survival. But what we've seen in our study, and now many others are starting to see, is that amputations are way down. And the reason for that, quite frankly, is based on these graphs on the screen. We're seeing vascular surgeons go down in number of procedures and a much more aggressive stance from endovascular physicians who are now going in with devices to be able to open up vasculature below the knee. What that has led to is a significant reduction in amputations. And what that means for folks like us running clinical trials is if that's your endpoint, you need to run a study with several thousand patients. So based on that, we sat down with some of our KOLs and started to really understand what the need is in this field. And this patient really demonstrates the paradigm of where CLI currently sits. It's a patient from the Cleveland Clinic who is our founding institution, but also a group with which we have close collaboration. You can see on the left, you had a patient come in with CLI. They had a minor wound on their toe, and based, I think, using here a angioplasty, they were able to open up the vessel and restore significant blood flow to the heel. And by doing so, they saved a major amputation, so major amputation being defined as one above the ankle. Unfortunately, if you look at this patient's wound about 6 months later, it has gotten much worse. And the real reason here is they've opened up the major arteries, but they've done very little for the microvascular flow, or the flow in the foot. And actually, a meta-analysis done, or analysis of registry patients done from the Cleveland Clinic showed that in a cohort of about 200 patients undergoing successful revascularization below the knee, that 45% of wounds remained unhealed after a year. And while the amputation rates were low in this group, I think somewhere in the mid-teens, what you also saw was 90% of those amputations occurred in patients with non-healing wounds. So what this has really done is shifted the paradigm of focus for CLI from no option patients at risk for amputation to those that are undergoing it below the knee revascularization that have poor flow to the foot and need help healing wounds. We approached the FDA with this and have now received approval for STOP-PAD. STOP-PAD is a 120-patient, double-blinded, randomized, placebo-controlled study in patients with advanced PAD and non-healing wounds that are undergoing below the knee revascularization. So ultimately, the way we like to think of it is we work alongside the peripheral interventionalists. We let them create the highway. We then create the side streets that will get the blood flow to the wound. We will be testing 8 and 16 mg doses, both of which showed preliminary signs of efficacy in our phase 2. And I think importantly, we will be repeat administering the drug once after the procedure and again 3 months later. Again, these are chronic progressive diseases, and we believe it's going to be important to stay on top of those with repeated administration of therapy. Our endpoint here will be wound healing, and we will be looking at that as a composite that includes both the improvement as well as the worsening of wounds across a 3 and 6 month period. One of the most attractive components to the therapy is it can be delivered anytime in 1 to 2 weeks after the revascularization procedure in about a 5-minute office visit, and it's 8 to 16 injections in the foot. We're near halfway done with the study right now. We're anticipating data in the middle of 2017, and that data will be informative in helping us move this towards later stage potential approval trials. Importantly, the FDA has improved its fast track designation, and we'll look to take advantage of that after the completion of the phase 2s in our discussions with them. So at a high level, we know that we have a drug in JVS-100 that has shown bioactivity in several preclinical and clinical studies. We are right now focused on our phase 2b STOP-PAD study that is enrolling at 20 sites in the United States, and we anticipate top-line data in mid-2017. We're approaching CLI really from a very different angle than a lot of companies in the space have, and we believe we can do that partly because JVS-100 provides a very attractive product profile in that it has affordable cost of goods and ease of delivery, scalability that allows you to deliver it in that specific setting. So I'm happy to answer any questions. We have a couple of minutes, or I'm happy to chat afterwards if there's any inquiries. Thank you.